very important publications in the field. And at the moment, he is a professor and group leader in Cima University, Cima or Cima? Cima University in Spain. And his research particularly focuses on two directions. One is to understand the mechanism of the development of Parkinson's disease. And second one, also very interesting direction, to develop gene therapy against curing Parkinson's disease. And for this purpose, he uses especially non-human primates. We are very excited and curious about his research and presentations, and thanks a lot.
Please. Okay, so good afternoon, everyone. A few things before getting started. First of all, to acknowledge Elhan and Sadiq for everything you have managed to prepare in such a short period of time. Thanks for your kindest invitation. It's a big pleasure to be here. Another thing I want to stress is that I am super happy to see so many young people here. As you may know, I'm here on behalf of IBRO. IBRO is a global neuroscience society merging together more than 100 societies all over the world. And the main commitment of IBRO is based on 4 pillars that are inclusion and diversity, gender balance, and geographical balance. So IBRO is there to support the careers of people in a very early stage. So most of you can benefit for the support of IBRO. We have a variety of programs that we support. You have some information in the registration desk. So I don't want to take all this information back with me. So please feel free to go ahead and just keep an eye on this. What else? I would also to thank the translator for the work you do. Thank you very much. And sorry to not be able to speak in Athari. I think for me it's already too late to get started with. So that's why, I have no other choice but to speak with you in English. I'm super happy to be here and for the friends and colleagues I have a chance to meet here today. Okay, so let's get started from the beginning. I will try to stay on time, but I cannot promise I will be on time. These are my disclosures, not anything special to comment. Just keep an eye also on another organization that is the European Brain Council. It's also, EBC is based in Brussels. It's an institution in charge of promoting neuroscience, advocacy neuroscience in European institutions. So if there is anything you need from patients association, pharma industry, scientific societies, the EBC is a good partner for everything you may need regarding the European institutions. What else? Well, we are using gene therapy. We are using gene therapy, and we are using gene therapy in 2 different ways. First of all, I'm working with non-human primates. That's, so some people will not understand this very easily. But, here, as the former speaker has commented, I think that nowadays it's hard to make difference between basic science, translational science, and clinical science. So all of us, we are here with the only commitment to find better medicines and better treatments for our patients. So science is not either basic, clinical, or translational. Science is just good science or bad science, and we always need to keep an eye on our patients, that this is why we work for. So that's why I use monkeys, because, in my field that is Parkinson's disease, don't worry, I'm not going to ask for how many people knows about Parkinson's disease. So in my field, a lot of people work with mice. They are doing very good job in mice. I have nothing against mice. However, when coming to develop new medicines, it's such a big difference between a mouse brain and a human brain that, not surprisingly, most of these treatments fail. So we are using gene therapy tools for the purpose of disease modeling. This is one part of my activities. When honestly speaking, my activities are bureaucracy. My people are the ones that are doing the beautiful job. We are using also gene therapy for therapeutic purposes. It's a nice field with great expectation. I think that the best is yet to come in the field of gene therapy. And we are creating better animal models, not just for fun. The way we create models And what we learn from the models is very helpful in an attempt to design what are the better targets that are going to be approached by gene therapy. So that's why we use models. So it has been discussed before. We have to recognize that most of the animal models we use are completely useless for the purpose we want to use these models. So even in the best case scenario, any given disease, no matter it's Parkinson's, Alzheimer's, or whatever, epilepsy, or whatever you have in mind, a disease is multifactorial, it's very complex. So this is, let's say, this mice is a mice with a real disorder. However, for the animal models we have, even in the best case scenario, we only reproduce a fraction of the pathology that characterizes the real disease. So if you design a treatment against this fraction of pathology, when translated to a real disease, this is not going to work. And humans, we are very stupid animal species. We always make the same mistakes time after time. So it was a nice party of people that are plenty of neuroinfluencers that get together in Germany quite a long time ago. They were, this is an initiative from Juan Carlos Lopez. At that time, he was the chief editor of Nature Medicine. So you get a call from the chief editor of Nature Medicine, you better go. So they spent a couple of days in Germany thinking about what are the main bottlenecks in disease, in disease-modifying therapeutics. And after a couple of days of heavy discussion, they came up with this list of 5 main bottlenecks. You don't need to be a neuroinfluencer to come up with this list. What they have stressed so much is that there is a pressing need to develop better animal models. So it is expected that with better animal models, we may learn more about the biology of disease, we can find better biomarkers, and we can, this is important, we can design better preclinical trials. And with a better preclinical trial, you may increase the chances of success of a later ongoing clinical trial. So for Parkinson's disease, there are plenty of gaps, that's for sure. We need models that reproduce better the signature of the disorder. So far, we know that cells that are producing dopamine die over time because they accumulate an intracellular protein that is alpha-synuclein that became misfolded and hyperphosphorylated. Of course, these neurons die over time in a progressive way. And by the time in which we first diagnose a patient, it's already too late. These patients have lost more than 50% of neurons. We need a model that is not acute. We need a model with a therapeutic window long enough to accommodate therapeutic interventions. Another thing that we often do not take in mind is that this model needs to be sustainable. So if I'm working with a non-human primate, and for my intervention, this primate needs, let's say, 5 years to develop Parkinson's disease, this is going to be unaffordable. So you need to be pragmatic and realistic in how long these animals are about to develop Parkinson's disease. And keeping in mind our patients, we need models that reproduce early stages of the disorders, but we also need models reproducing late stages of the disorders because we have patients of both types. Okay, so we have 2 types of models trying to enhance the expression of tyrosinase. We are using a gene that is human tyrosinase that is the enzymatic precursor of neuromelanin. You will understand in a minute. And we have also models intended to enhance the expression of alpha-synuclein. Well, this is the real disorder. As mentioned before, that humans, we are a stupid animal species. We are trying to model a disorder without the appropriate knowledge of the things we want to model. So Parkinson's disease, neurons that are about to disappear are pigmented, are full of neuromelanin. It's a dark pigment that is intracellular. This is why the substantia nigra is called substantia nigra. It makes reference to a dark pigment. Otherwise, might be called green, substantia green or substantia red. No, it's called nigra because it has a lot of neuromelanin. And only humans, we have neuromelanin, humans and non-human primates. Only humans are suffering for Parkinson's disease, so there might be a link between neuromelanin and Parkinson's disease. Of course, in patients, we lose neurons that produce dopamine. And as a consequence of this neuronal loss, we also lose pigmentation over time. And what a neuropathologist needs to see when looking behind the microscope is a number of intracellular inclusions called Lewy bodies. This is the characteristic hallmark of the disorder. Lewy bodies are intracellular inclusions made of alpha-synuclein. This is one neuron that is pigmented. I'm not sure you can see the grains of neuromelanin. They are very thin here. Lewy bodies can be either single or multivesicular. And sometimes there is another smaller inclusion. You can see here, here, and here. That is at the level of the nucleus, and it's called Marinesco body. Marinesco bodies are an indirect indicator of advanced aging, and it's typically found in the substantia nigra. So this is, in brief, this is what we want to reproduce in a good animal model. We want to reproduce these neuropathological signatures that typically characterize Parkinson's disease. We are not looking for a symptomatic model, we are looking for a neuropathological model. And this is our strategy. As I mentioned before, we perform stereotactic injections of adeno-associated viruses coding for human tyrosinase in the left substantia nigra. We perform sagittal ventriculography and MRI navigation to get into the substantia nigra with an antivirus that is used as a control in the right substantia nigra. So once the neurons are intraparenchymally delivered, then transduce the neurons. These neurons start to accumulate neuromelanin over time. At some point, these neurons start to exhibit intracellular inclusions. And once these neurons die, neuromelanin goes extracellular and start to activate a number of microglial cells that creates overall a pro-inflammatory environment that perpetuates and accelerates neuronal death. Well, this is just a macroscopic view of two cases. This is the brain of two monkeys has been delivered in the left substantia nigra, the neuromelanin coding vector. This is the substantia nigra. This is the midline. This is the substantia nigra. The hippocampus is here, third ventricle, and this is the thalamus. This is rostral substantia nigra, going back to the caudal part of the substantia nigra. This is the lateral geniculate body for reference here. This is also an animal with a follow-up of 4 months. So this animal had been sacrificed 4 months after the injection of the viral vector coding for neuromelanin. You can see that the entire substantia nigra became transduced and became pigmented. If you wait a little bit longer, I hope you can appreciate that the substantia nigra still is pigmented, but there is, compared with 4 months, it's a clear loss of pigmentation reflecting dopaminergic cell death. Okay? What next? Well, this is neuromelanin. In 4 cases, this is the substantia nigra. This is neuromelanin without any type of counter stain. This is counter stain with neutral red. We can use a 2-layer algorithm to count pigmented neurons versus non-pigmented neurons. We can calculate it here. I don't want to enter into too many details. This is the type of lesion we are about to induce in the substantia nigra. Even if you are not that much familiar, you can see the substantia nigra has a clear loss of neurons compared to the right substantia nigra. This is the caudate and the putamen on the injected side. On the contralateral side is the control of 2 animals here, 2 animals here. I hope that at least in these 2 cases, you compare left and right putamen or left and right caudate. You can see there is a reduction in the number of dopaminergic terminals. This is a loss of stain reflecting neuronal death. And this is probably one of the most important findings of the paper. This is just some confocal microphotographs. This is neuromelanin. This is immunostain for tyrosine hydroxylase. So we can demonstrate these are dopaminergic neurons in the substantia nigra. And these are Lewy bodies. Lewy bodies are positive for P62, for total alpha-synuclein, for predigested alpha-synuclein, and for phosphorylated alpha-synuclein. This is the first description of Lewy body pathology in non-human primates. And the important issue is that this synuclein that is hyperphosphorylated is made 100% of endogenous alpha-synuclein. We don't have enhanced alpha-synuclein. We just simply enhance neuromelanin accumulation. Well, this is the microglial reaction that is mostly concentrated in perivascular spaces, exactly the same as in human brains. And this is just to show you the microglial reaction. These are EVA1, that is a marker, a pan-microglial marker, CD68, that accounts for activated microglial cells. And this is neuromelanin, so you can see these neurons have digested extracellular neuromelanin. Another thing that I don't want to enter into many details is that this neuromelanin can spread to the frontal cortex. This is taken from the superior frontal gyrus. You have a layer 5 pyramidal neuron that is about to receive a dopaminergic terminal with a terminal button here, making a contact on this layer 5 pyramidal neuron. This is a Lewy body that is just located in the postsynaptic element, because we know that alpha-synuclein can spread in a prion-like fashion by taking advantage of anatomical throughputs. Okay, so basically, in brief, we managed to create a very nice neuropathological model of Parkinson's disease in non-human primates that reproduce all the neuropathological signatures of the true disorder in non-human primates. Okay, so next, it's not only about the model, but what we learn from the model. Well, the substantia nigra, this is the substantia nigra, this is the red nucleus, this is the midline, this is another, the same animal, substantia nigra, red nucleus, and the midline. The substantia nigra is made of two different types of neurons. They are all dopaminergic, but they are distributed in what we call two different tiers. We have the dorsal tier, that is positive for calbindin, is here, and the ventral tier, that is calbindin negative. So we have markers for dorsal and ventral tier with calbindin, and quite the opposite if we stain for this marker that is aldehyde dehydrogenase type 1. Aldehyde dehydrogenase is negative in the dorsal tier and is only positive in the ventral tier. So if you go to higher magnification, although it can be seen here, you can see that most of the pigmented neurons are located in calbindin-free areas for some reason or another. If you go to high magnification, these are neurons in the ventral tier, plenty of neuromelanin inside, even a Lewy body can be seen. And these are neurons of the dorsal tier that are positive for calbindin, with not that much of neuromelanin, and because they don't have enough neuromelanin, these neurons very rarely display a Lewy body. If we quantify Lewy bodies, what you have in green are Lewy bodies in the ventral tier. What you have in purple or pink or magenta are Lewy bodies in the dorsal tier. And this is the quantification carried out in 4 animals. You can see that more than 98% of Lewy bodies are located in the ventral tier. And this is what we learned from the model. So in the substantia nigra, we have two different types of neurons. Neurons in the dorsal tier that are positive for calbindin. These neurons exhibit very little amount of neuromelanin. And because they don't have that much neuromelanin, they never display Lewy bodies. However, neurons in the ventral tier are much more heavily pigmented because these levels of pigmentation, these neurons are the only ones to have a chance to develop a Lewy body, and that's why these neurons die. And this is in full keeping with the available literature. We know the neurons in the ventral tier that are those innervating the posterior putamen, the postcommissural putamen, are much more vulnerable than neurons in the dorsal tier that are more resilient. So in the end, dopaminergic vulnerability is the result of a synergistic effect between levels of calbindin, levels of neuromelanin, and presence of Lewy bodies. All these things together create like a perfect storm that in the end defines dopaminergic cell vulnerability. Okay. The field of gene therapy is quickly evolving at a breathtaking speed. There are a lot of people looking for capsids that are engineered to penetrate the blood-brain barrier. By going this way, you can induce gene expression in the CNS without the need to perform stereotactic surgery. So there's a company in Cambridge called Handl Therapeutics that has developed a capsid variant of AV9 called P31 that once delivered systemically has a very nice capability to penetrate the blood-brain barrier and to transduce the brain. So we have mounted the tyrosinase gene into this BBB penetrant capsid variant of AV9. We have a cohort of animals with 1 month post-delivery, another cohort with 4 months. Now we have 3 more cohorts at 6, 8, and 12 months. And you can see this is the mouse brain. Both the substantia nigra and the VTA are pigmented even to the naked eye. With 1 month of injection, with 1 month of cerebral time, you can see small amounts of neuromelanin in the VTA, in the substantia nigra and VTA. If you wait a little bit longer, and this is 4 months of follow-up, you can see the amount of neuromelanin is being increased quite heavily, both in the substantia nigra and in the VTA. These animals have neuromelanin in the pioneering neurons and have markers, typical markers for Lewy bodies. So in brief, you don't need to perform, you are dealing with mice, you don't need to perform stereotactic surgeries. You can induce pigmentation just by going systemically with this variant of AV9. I mentioned before, we also need to develop models. So how was that time? Well, we need to develop models mimicking later stages of Parkinson's disease. And this is a big controversy in the field of clinical movement disorders. We have 3 different entities, that is Parkinson's disease with dementia, and dementia with Lewy bodies. And these categories depends on the clinician you are approaching. If you are asking for a neurologist about this, you say, okay, these are 3 different entities. One thing is Parkinson's disease, a different issue is Parkinson's disease with dementia, and a different presentation is dementia with Lewy bodies. However, if you talk to a neuropathologist, you will likely retrieve the answer that these are the same disorder, but in different stages of progression. But this is a never-ending discussion. But it doesn't really matter. My point is that, these patients became demented in later stages of Parkinson's disease and dementia with Lewy bodies because they exhibit a cortical synucleinopathy. So synuclein is no longer in the nigra, but they have also Lewy bodies in the cerebral cortex. And we also need models trying to reproduce dementia with Lewy bodies and Parkinson's disease with dementia. We need animal models with synuclein in the cerebral cortex, in brief. So this has been approached by taking advantage of an AV9 coding for mutated alpha-synuclein, the most common mutation, that is A53T. So we know that AV9 is a capsid variant, is a serotype, AV9 is a serotype that has the ability to spread in the retrograde direction in the brain by taking advantage of the underlying brain circuits. So it behaves like a retrograde tracer. So once delivered in that case, into the putamen, AV9 is going to spread in the retrograde direction, so you can induce expression of any given gene in every single area that is connected to the injection site. So neurons in the cortex that are created to the putamen through the corticostriatal pathway, neurons in the thalamus that are innervating the putamen through the thalamostriatal pathway, and neurons giving rise to the nigrostriatal pathway. These neurons, all of them, start to express the genetic payload that is encoded by AV9. So here what we did is we deliver AV9 coding for mutated alpha-synuclein into the putamen. This is the way we perform surgery without navigation. This is just a sagittal ventriculography. So in brief, we deliver an X-ray contrast in the lateral ventricle in the frontal pole, so we can delineate the entire ventricular system. So for instance, this oval area here is the thalamus that is surrounded by the third ventricle. So by going this way, we can allocate the position of the anterior and the posterior commissure. This defines the ACPC plane, and this is the 0 point from which we start calculating stereotactic coordinates. So we deliver 25 microliters of this AV at the level of the anterior commissure, another deposit 1 millimeter rostral to the anterior commissure, another deposit 1 millimeter caudal to the anterior commissure. So So by going with this way, this is the putamen, the internal capsule, the anterior commissure. You can see a little bit of the caudate nucleus here. This is another animal, the putamen, internal capsule, anterior commissure, a little bit of the caudate here. So you can see we have, by performing 3 injections of this AAV, we have filled almost the entire extent of the putamen. This is the extent of the putamen area being transduced with AAV. It's roughly about 70% of average. It's quite a nice coverage of the putamen. And by going this way, then we can perform a biodistribution study. A biodistribution study is made of 3 different readouts. We need to know the total number of neurons that are expressing alpha-synuclein all over the brain of the primates. We need to know which is the rostrocaudal distribution of these neurons. We need to know which is the accurate location of these neurons. So number, rostrocaudal distribution, and location are the 3 main readouts for any biodistribution study. And in order to quantify neurons, I can tell you the non-human primate brain is quite a large piece of tissue. If we just want to count neurons by relying on stereology, it will take weeks to count just one single section. This is not affordable. So we are using AI, a dedicated AI algorithm from a company from Finland that has a 96% of accuracy compared to traditional counting methods, but saves an incredible amount of time. So in order to quantify neurons, I can tell you in the earlier times, to quantify the new number of neurons in the substantia nigra in just one section can take about 1 hour and a half. With this type of AI algorithm, it takes just a matter of a couple of seconds. So it saves an incredible amount of time with a very good accuracy. So this video is not going to display, I'm sorry, but here you have the supramarginal gyrus of the parietal lobe. You can see several neurons, I hope you can see. These neurons are all located in layer 5 and are neurons that are expressing alpha-synuclein. So we know that all these neurons are corticoputaminal projecting neurons because they are innervating the putamen. This is where we have put the virus. And this is the readout of the algorithm. Every single dot is one of those neurons. You can see all these neurons has been quantified. There is no neuron left without being quantified. And the accuracy is very nice. I'm sorry that the video is not going to run. So this is the first readout, number of neurons. You have different regions in the cerebral cortex and subcortical locations here. Well, most of these neurons are located in the prefrontal gyrus on both animals. This is superior frontal gyrus, anterior cingulate, posterior cingulate, insular gyrus, substantia nigra, and so on. And this is the contralateral corticostriatal projection, superior frontal gyrus, precentral gyrus, and so on. So number of neurons, then distribution in the rostrocaudal space. So for instance, this exercise has been made. You take a look at the postcentral gyrus in the parietal lobe. So it's made of 9 millimeters. Each cube, each square is made of 1 millimeter. So this postcentral gyrus have a given number of neurons. Let's see here. This posterior, sorry. This is the posterior. This is the number of neurons in the posterior gyrus. But now we want to see which is the rostrocaudal distribution. You will see that a given number of neurons is not equally distributed. So most of these neurons are located in the more rostral part of the posterior gyrus, posterior central gyrus. And this is location. These are maps of 3 different levels of the brain of one animal and the other animal. 6 millimeter rostral to the anterior commissure, at the level of the anterior commissure at 9 millimeters caudal. Every single point is just one neuron. You can see the distribution is specific of layer 5, because we know that in layer 5 is where all corticostriatal neurons are going to be located. This is just some pictures, and this is the anterior cingulate left and right, superior frontal gyrus left and right, prefrontal gyrus, supramarginal gyrus, and sometimes you can even see Golgi-like label neurons expressing alpha-synuclein. And this is the substantia nigra, counterstandard with Basal, to better define the cytoarchitectural boundaries. This is the red nucleus, this is the midline, this is the substantia nigra. And you can see in the substantia nigra, roughly half of the neurons of the substantia nigra are expressed in alpha-synuclein. And this is the extent of tyrosine hydroxylase terminals in rostral and caudal parts of the caudate and putamen, and in the substantia nigra. And this is what we have modeled. This is my team. This is my team. These are our grant funding agencies. These guys are the guys that are doing the nice job. I'm just the bureaucrat of the team. And that's it. So it was a real pleasure. I hope I didn't extend too much out of time. And now you have any questions, please feel free to go ahead.
Thank you so much for the talk.
Hi, thanks a lot for great presentation and amazing data. I had two questions, quick ones. One, you had to use P31 in the plasmid for the delivery through the BBB. I was wondering what's the efficiency of it compared to P75 or transferrin, if there was a particular reason to choose this one. And second reason for the segmentation that I understand it's out of time. That's why when I studied Alzheimer's in the entire mice brain models, I was also quantifying A-beta plaques, for example. And there, for example, one advantage is that there is very good clearing techniques that you can quantify in the entire brain without even going to segmentation, keep the intact brain. I was wondering how much it has developed in the macaque research field to use this field.
Okay, so for the first part of the talk, this BBB penetrant variant of AV9 called P31 has been developed by what they call capsid engineering. So in brief, there are two different ways of generate new AV9 capsids. You can go one way with the so-called direct evolution, or you can allow these viruses to mutate in a natural way, or you can use artificial intelligence to generate new capsid variants. This is capsid engineering. Capsid engineering is much more, let's say, robust, I will say, in terms of readout. The problem with capsid engineering is that in the end, you get what you want. So, for instance, this AV has been designed against a receptor that is only present in the BBB of mice. It doesn't work in rats, it doesn't work in non-human primates, and for sure it's not going to work in humans. So it's very good for, let's say, basic research, but if you want to develop an AV capsid variant with a therapeutic transgene, I think that capsid engineering is not the way to go. Regarding clearing of mouse brains versus non-human primate brain, clearing is a technique that, in brief, you can, I would say, dissolve the brain, but the labeling remains there, so you can create like transparent maps in 3D of one entire brain. And this is very good in order to get an overall 3D view of what is inside the brain. However, for a non-human primate, it's much more difficult because these are, compared to a mice, these are much bigger brains. So our advantage is that when we deliver the neuromelanin coding vector in the substantia nigra, in this it's coding for tyrosinase, it's not coding for neuromelanin. So we may enhance tyrosinase expression in any given neuron in the brain, but it doesn't really matter because in the end, we are only to induce pigmentation in dopaminergic neurons because neuromelanin is a subproduct of dopamine oxidation. So you need to have the machinery of synthesis of dopamine to become pigmented. So in that case, in this AV, in this particular AV, cellular selectivity is not driven by the promoter, for example, it's driven by the encoded transgene.
I'll also just ask a quick question. So from coming very naive, very ignorant question. So it seems like one of the main problems is the alpha-synuclein, right?
Yep.
So it gives to death. How far are we from designing some sort of enzyme that we can deliver that can solve?
Well, yeah, it's a very good question. It's not a naive question. I can tell you there's a very heated debate going on in this focus. Synuclein, we know synuclein is the main constituent of Lewy bodies, that's for sure. However, we are not that sure to what extent Lewy bodies are pathogenic or not. Some people will claim neurons are about to die because of alpha-synuclein accumulation. And so people are claiming just the opposite. The Lewy body is a sophisticated way to package alpha-synuclein to not to be harmful to neurons. Now, the main problem we are now, and it's not that easy to elucidate, is that if you take a look at the brain, at postmortem brain samples of humans suffering from Parkinson's disorder, the real disorder, not a model, but the real disorder, it's almost impossible to see a patient with Alzheimer without with A-beta, with amyloid plaques and tau tangles, without Lewy bodies. They have everything. And the same holds true for Parkinsonian patients. You take a look at the substantia nigra of a patient in later stages of Hoyne-Yahr, let's say 5 or 6, you will not be that much surprised to see not only Lewy bodies in the substantia nigra, but also tau tangles. So there are some provocative papers coming in blaming tau for the cellular death of dopaminergic neurons instead of Lewy bodies. And the debate is there. It's going to be there for a while because it's not that easy. So this type of neurodegenerative disorders in late stages, they are often seen as multiple proteinopathies. And it's hard to distinguish to what extent the blame is on tau, the blame is on Lewy bodies, or the blame is on amyloid. It should be a mix of everything.
Thank you so much. Well, if you have no more questions, I will ask you to give a very warm applause for our teacher for being here. So you might have realized